Okay, well, welcome everyone to today's Safer World by Design webinar. And today we're going to focus on applications of genotoxicity testing for risk assessment. We're going to have two talks, and then we'll have a discussion. Keep in mind you can submit questions at any time through the panel on the GoToWebinar in the question box, and then we'll work through, I hope, some good questions and discussion after the two talks. So to start with, the first speaker will be Marc Audebert, and he's going to discuss the use of human cell lines with different bioactivation capacities to determine genotoxic mechanisms of action. And I'm going to hand over to you, Marc, for what I'm sure will be an interesting talk. Over to you.
Hi, everyone. Thanks a lot to invite me to present you some of my work. So I'm Marc Audebert. So I'm working in Toulouse in the south of France, and I'm working for INRAE and also co-founder of a Preditox company in France. So first of all, why use genotoxicity screening? The first point is that carcinogenicity assays are mainly done in mice and rats, and genotoxicity could be done in different cell lines, notably human. And the costs are not the same, much more million for carcinogenicity compared to thousands euro for genotoxicity assay. Time are not the same. It's more than 2 years to have the final results of carcinogenic assay, and maybe 2 weeks for most of the genotox. For the 3R rules, clearly carcinogenic assay are a problem, and genotoxicity are much more 3R compliant. And for the regulation, now more and more genotoxicity are included in implementation, and there are also some comparison for predictivity, because if we want to have an idea of the carcinogenic potential of compounds, it could be much more complicated when we check for a human, and much more with human cells. So sorry. So increased world needs in vitro compliance with new legislation like a REACH program and the development of new genotoxicity. But there are some limitations of the current applied genotox assays. So there are some intra-species differences for metabolism on DNA repair, like for in rats and rodents compared to human or bacteria. You need hassle-through screening. You need to be sure that you are not false positive with cytotoxic compounds. And more recently, you have to determine the genotoxic mode of action of your compound. So to know if it's an antigen or a clastogen. So you have to know that DNA could be damaged by a lot of things. It could be single-strand break, it could be oxidative stress, it could be cross-link and whatever. And when you have adjacent against strand break, you have mechanistically a double-strand break. However, if you trap topoisomerase, or if you have blocked transcription or replication, whatever the DNA damage, or you interfere with DNA repair, in this case, at the end, you will have induced double-strand break. So you have to keep in mind that double-strand break could be induced by a large class of DNA lesion when cell replicate and transcribe normally. So here it's a picture of the chromatin. So as you know, you have the DNA at the start, and you have the nucleosome. It's a protein that the histone core that wraps the DNA. And in this histone, you have different histone H1 and H2, H2A, H2B, but also you have the variant named H2X. And when this histone, when you have a double-strand break, in this case, this particular variant of histone H2X will be phosphorylate there. And you can look by aminofluorescence staining apparition of DNA damage in the cells. So this phosphorylation is very rapid. This histone is present in all cells, and you have with this immunostaining a quantitative response to DNA lesion. And moreover, it has been demonstrated in precursor cells in vivo. So you could easily detect HDX phosphorylation by immunohistochemistry, and for more than 10 years now, I use this biomarker for genotoxicity. As you can see here, some of the paper we have done with different type of contaminants. We have like pesticide or bisphenol, mycotoxin, pH, and so on and so on. However, when we check for this biomarker, we could not easily distinguish antigen from clastogen. So we decided to check for another biomarker named histone H3 that is phosphorylated at 710 when cell goes through the cycle and enter mitosis. And when you have antigenic compound, generally you force the cells in mitosis, so you have overactivation and overphosphorylation of this histone H3. And moreover, some could be also inhibit the phosphorylation. So we have proposed a new strategy for genotoxicity screening and named gamma-HDX phosphorylation or a pre-screen test on human cells. So if you have an unknown compound, you can test it on FG2 or HEPA-IgG. And if you have a decrease in the phosphorylation of these two histone, or you have a clear decrease of the viability, because we have also checked for viability in parallel of genotoxicity, in this case, your compound is just classified as cytotoxic. If you have a great increase or decrease of phosphorylation, in this case, you have an antigen, and you could have a two mode of action of antigen. And if you have an increase of gamma-HDX, in this case, most of course, it could be a clastogen mode of action compound. And if you want to be sure of the results and to have other indication of what the bioactivation, you can screen on two other human cell lines. It's a colon cell line there and a kidney cell line. This one was able to bioactivate compound, and this one was totally devoid of CYP and other phase 1 and phase 2. If you have an antigen, you will have the same type of results. If you have a direct clastogen like oxidative stress or MMS or alkylic agent, in this case, you will have the same results as in FG2 or HEPA-IgG cells. But if you have a different result between the two cell lines, and that only bioactivation cell lines could have clear results, it could be need bioactivation. It could be the case notably for benzoaparin or some of this type of compound that need phase 1 and phase 2. So with this new strategy, you have a clear quantitative and qualitative indication of on the mode of genotoxic action. You can see that it's an antigen, a clastogen, and if it needs or not bioactivation. So here is a meta-analysis we have done 2 years ago with two biomarkers. So we select 27 publications, a lot of different cell lines, Texas, FG2, HEPA-IgG, and whatever. Different techniques have been used by different labs, like flow cytometry or Intel Western, like in my lab, or eye content screening. There are one Intel about the history, more than 300 different compounds with different class, antigen, clastogen, or non-genotoxic, and different mechanisms of action. When we check for sensitivity, so to be sure that you are not false positive, we are more than 95% of sensitivity. For specificity, we have 88%, and so an overall predictivity of 91%. But when you check the different compounds, so here the 4 compounds are classified in 5 carcinogens. So in my mind, maybe gamma-HDX, this new biomarker, could be able to determine carcinogenic compounds that could be not determined easily with other genotoxicity. So in this case, you have a sensitivity of more than 98%. For the specificity, we have to keep in mind that 8 compounds have been tested in only one study and at only one concentration. And if we exclude this compound, in this case, we have more than 91%. And at the end, we have an overall predictivity and accuracy of this two biomarker of 95%. So if we compare with more classical genotox assay like macronuclei, you can see here BMD 50 or in vitro macronuclei compared to here it's log 10 for phosphorus 3. You can see, and here you have the same for clustergenic with gamma-HDX compared to macronuclei. You could see a very, very high correlation between the macronuclei and gamma-HDX phosphorus 2. If we compare with here, I'm not sure it worked well. Here. Here you can see a comparison of different in vitro genotox assay. So I put just Comet macronuclei, reporter assay like blue screen or tox tracker. And here you have gamma-HDX phosphorus 3. For the DNA damage that can be detected, it knows that for Comet you need 10 to 100 different strand breaks to be detected. Macronuclei, you only check for DNA in cytoplasm, and reporter says not directly DNA damage you can check. And here with gamma-HDX, we know that we could be able to detect as low as 1 strand break per cells. It's in vitro, in vivo concept biomarker. It's not the case for notably reporter 1. Cells, you could use whatever the cells you want for Comet, macronuclei, or gamma-HDX phosphorus 3. In a reporter cell, you could not choose the cell line you have. You have a very high throughput with reporter and gamma-HDX, and now also for macronuclei with FACS. For genotoxicity, mode of action discrimination is the only assay that could do it. And we have very high level of predictivity compared to the other one, and very great reproducibility and availability. And at the moment, we have some discussion for this validation at a different level. So when you can check for SCCS is now in the last revision, and it could be used for ECA and CSO or as a user weight of evidence approaches. And I think maybe we could have to discuss to go to OECD guideline like for macronuclei. So the conclusion take of nature, it's a gamma H2, it's a global DNA biomarker like macronuclei. It could be performed in every cell type and whatever in suspension or whatever cell cycle or whatever. It could be is the most predictive in vitro assay at the moment, and also it's the only one test that could discriminate very easily cytotoxic antigen and clustergen in one single assay. So thank you for your attention, and of course you can check me by email or have a look at the Predictox website. Thanks a lot.
Well, thank you, Marc. I'm now going to hand over to Dimitrios, who will be our second speaker. And let's see, Dimitrios, if you can take the screen for us.
Can you see my screen?
Yeah, so now we have your title slide,
Thank you very much.
and
Great.
off you go.
Okay. Good morning, good afternoon, everybody. I'm going to talk a little bit about the Ames MPF, which is a miniaturized microplate format version of the Ames assay. The menu is just briefly introduce the Ames test and its importance in the risk assessment world, what the Ames MPF is, and its advantages, what efforts have been done to validate it, and some real-life examples of how it can be used in a regulatory context. The Ames test, otherwise known as the bacterial reverse mutation assay, has had a very broad impact over the last tens of years. So as you can see here, it's mentioned in the ICH guideline on the assessment and control of DNA reactive impurities in pharmaceuticals, as it is for the interpretation of the results of data from pharmaceuticals intended for human use. And the EFSA suggests it as one of the 2 in vitro tests for the first steps of assessing food and feed safety assessment, and for plant protection product active ingredients is basically the only assay that is used by a plethora of countries. This is also the EU REACH. Basically, the Ames test is used constantly by a lot of regulatory agencies as a primary threshold. In some cases, also basically the only experimental assay, because it's been shown that it has a fairly good correlation with carcinogenic data, and so the Ames test with an assay performed on mammalian cell lines normally is pretty good in detecting what could go wrong. As a matter of fact, Harold Zeiger in a 2019 paper called it the test that changed the world, which sounds great, but it's not very far from reality. And the OECD guideline 471 defines how the protocol should be performed. Very briefly, mutagens are assessed using bacterial reverse mutation with deficiency mutants. So Salmonella typhimurium and E. coli strains have been developed so that they cannot grow in the presence of histidine, in the absence, of histidine or tryptophan, respectively, due to base pair substitutions of frame shift mutations. So if you plate these auxotrophs on amino acid deficient medium, you will have that most of them will die, apart from some prototrophs to reversion. So mutations that can be spontaneous, as it would be the case in the solvent control, or sample induced if your sample is mutagenic. So after a couple of days, you count how many colonies you have in your plate, and by testing in the presence and absence of a S9 liver fraction, which mimics the mammalian metabolism, you can sort of also make up for the bacterial limited enzyme equipment. So chemicals as benzoalphaperine, which is not mutagenic, as opposed to its metabolites, can turn out to be positive. And it's remarkable that this assay performed in bacteria performs quite well in predicting what the effect in humans could be. The bacterial reverse mutation assay in the Ames2 Ames MPF format is performed in multi-well plates. So it's liquid, low volume, and has a colorimetric readout. So instead of having, for example, 8 Petri dishes, you have a single 384 well plate. It contains an amino acid deficient bromocresol purple containing medium as an indicator medium. So the bromocresol purple is purple, as the name says, and when no mutagenic activity occurs, the bacteria will remain auxotrophs, they will quiesce or die, and the pH will remain the same. When revertants occur, one should know that they are metabolically active cells, so they will proliferate and synthesize organic acids, leading to an acidification of the medium, making that the medium will turn yellow. So you can really count, instead of counting colonies, you can count how many yellow wells you have. There are a lot of advantages over the Ames test performed in Petri dishes. You need less sample, 4 times reduction of the samples, which is important when you have it in limited quantities or to assess genotoxic impurities. The throughput is dramatically increased, which means that more samples can be tested simultaneously using multi-channel pipettes. And importantly, it's also automatable, not only the experiment itself, but also the plate reading. And the plate reading normally takes a lot of time for the pre-incubation Ames test, the Ames test in petri dishes, which is, this is dramatically reduced, as is other hands-on steps for the Ames MPF. And when you have to count hundreds of colonies, it's all fun and games until a certain point. So this step, which is also error prone, is favorably affected in the Ames MPF format. Importantly, there have been publications that have shown that direct comparison of the Ames MPF assay and the Ames test in petri dishes show that the sensitivity, meaning the responsiveness at low concentration levels, is better for the Ames MPF. So you will detect weak positive and equivocals, but also positive chemicals at lower concentrations in the MPF compared to the Ames test in petri dishes. Finally, the kit is standardized, which means that everything is performed with bacteria, strains, and substances that are certified to have passed the quality control. Importantly, there are also environmental advantages. So you have to use less liver S9 homogenate. This in the context of the 3 Rs, reuse, reduce, and recycle, means that you will have to sacrifice 13 times less animals compared to the Ames test in petri dishes. And there is also a dramatic reduction of the plastic waste, because instead of 8 petri dishes, you will only have a single 384 well plate, which means that instead of a small mound of petri dishes, you will have pretty much a handful of 384 well plates. And this also has an important effect on the costs of disposal. What is the acceptance of the Ames 2 and Ames MPF, since we've seen that the Ames test is accepted pretty broadly? One should say probably that the Ames MPF normally is used for the screening phase and for environmental assessment. So having a look at some documents from the regulatory agencies, one should say that the Ames MPF, or the fluctuation method, is mentioned in the OECD guideline 471, and it's actually suggested for by the ICH M7R1 in some conditions. For example, for impurities that are not feasible to isolate or synthesize, or when compound is in limited quantities. In this case, the bacterial mutagenicity testing, so the Ames test, can be carried out using a miniaturized assay format with proven high concordance to the ICH compliant test. Also, a microfluctuation specific ISO document standard has been published for the determination of genotoxicity of water and wastewater, which leads me to my next step, which is how concordant is the Ames MPF compared to the Ames test, because it's actually important that there is a proven high concordance. And recently we published a paper at Xenometrix focusing on a selection of the chemical from the recommended lists of genotoxic and non-genotoxic chemicals for assessment of the performance of new or improved genotoxicity tests. This is basically a list which seems to have been devised for this type of analysis. The first paper came out from the as a follow-up of an ECVAM workshop in 2008, and the list was updated in 2016. There's a list here. This is not an eye doctor visit, so you don't really have to read the individual chemicals, but the important thing is that you might see that some of them are maintained in both lists. Some of them were added to the second list, some of them were kept but with a different classification, and some of them were dropped. We selected 60 chemicals from this list and tested them in up to 5 strains. because the Ames test, I should mention at this moment, it can be performed in different strains that have been developed to identify slightly different frame shift or base pair mutations, which in turn gives you a fingerprint of what the mutagenic effect of your sample might be. What we did, we tested him using, also in collaboration with Preditox, who performed some of the tests, and compared our data with the published data from the NTP database and from other publications where raw data were available. And we noted that there is an excellent agreement. So one should keep in mind that the Ames test has been published to have an interlab concordance of 87%. So 87% is how reproducible the Ames test is in some cases. And we can see here that the concordance of the Ames test results published in the Ames MPF was higher than I think that here the lowest value is this 87.8%. And this is quite important. So we are in a ballpark which is as similar as the Ames test in petri dishes. And importantly, many of the discordant results could be clustered, both for the Ames test and the Ames MPF, meaning that in some cases, we have reason to believe that there was some company-specific purity issue or lot-specific purity issue, which in follow-up studies that we performed after this analysis was performed, we confirmed to be true. So probably these concordance values is while being very, very good, is still an underestimation of how well the assays are similar. And this is in the context of a lot of validation studies that have been performed with different assays. The Ames2 and the Ames MPF differ for the strains used. But as you can see here, using chemicals, company-owned chemicals, coded chemicals for a multi-center study, environmental samples, the accordance is always pretty good. In any case, normally higher than the published Ames test interlab accordance. And we also have a couple of direct comparisons, which is the proper way to compare the two assays, meaning the same overnight culture of the bacteria, the same sample preparation. And in both cases, the LEC values, the lowest effective concentration values, are lower for the Ames MPF, indicating that you need lower concentration to have a positive signal for a positive sample in the Ames MPF. What samples, what users? Labs in more than 20 countries, including pharmaceutical and cosmetic industries, EU and national governmental agencies, CROs, military, use the Ames MPF to test pharmaceuticals, chemicals, cosmetics, pesticides, environmental samples, food packing materials. So you can see that the risk assessment can be performed in different assays, in different contexts of types of chemicals. And here you can see only some of the Ames MPF users in the world and specifically in Europe. What about any specific example of how the Ames MPF can be used for regulatory purposes? There are more than 100 publications listed on the Xenometrix website. As you can see here, there's some, for example, from the US Army, which states how the regulatory decisions involving US EPA and FDA can be used using these Ames test assays. For example, here there's a publication on a major impurity for drugs. And among the studies, toxicological studies related to impurities, two of them, not only this one, but additionally two more use the Ames MPF. And also you can use the Ames MPF for particulate matter, for example, for biodiesel fueled engines. There are a handful of papers on this. More specifically, a pharmaceutical laboratory reported that they've been using the Ames2 since 1995 with a very good internal validation study indication of agreement with the Ames test. And in this 2007 publication, they reported that they identified no false positives, but there were a few false negatives, which eventually they understood that they were basically chemicals that were positive in a specific strain that they were not using. So since they implemented it using an Ames to Ames MPF hybrid, they have a 98% correlation with the regulatory aims. Normally they use it in the research stage, and development stage they use the regulatory aims test. Here I will just very briefly state that the pharmaceutical company that we've been that has been using the Ames MPF has registered with the FDA compounds, and has registered also with the French agency compounds that were based using data obtained with the Ames2 assay. And finally, a real-life example for environmental samples is what happened in Glarus. A while ago, they performed some tests with pilot samples from the wastewater treatment plant in Glanderland, which is in Glarus here in Switzerland. It's this canton here, southeast. And they used the Ames MPF 98100. So they used 2 strains. And the place is pretty beautiful, so we'd like really to keep it that way. So they, for the wastewater treatment plant, they tested how the samples performed before ozonation, after ozonation, and after granular activated carbon filtration. And as you can read from this text, they observed that there was a very high mutagenicity after ozonation, which is this plot here. The samples in the Ames MPF proved to be slightly positive before ozonation. After ozonation, there was a dramatic increase in the mutagenicity of the sample, which was completely disrupted after the granular activated carbon filtration. This led to the decision by the Swiss Federal Office for the Environment and the wastewater treatment plant operators to install a granular activated carbon step. This is just to show how results of the Ames2 and Ames MPF can have an impact on real life, both for pharmaceutical oriented chemicals and for environmental samples. So summing up, the Ames2 and Ames MPF is a miniaturized microplate liquid format of the Ames test, or the bacterial reverse mutation assay, with several advantages over the regulatory Ames, including a higher throughput, less hands-on time, especially for data analysis, a dramatic reduction of the sample and S9 mix needed, and generally a higher sensitivity in terms of limit of detection. Several validation efforts, at least 2 are currently ongoing, indicated the Ames MPF is a reliable alternative to the regulatory Ames. And albeit to a limited extent, the Ames2 and Ames MPF are to some extent accepted by regulatory. So one should probably double check first, but in any case, a lot of Ames MPF users do is they use the Ames MPF for a primary screening, and then for the eventual validation by regulatory, they use the GLP Ames test in Petri dishes. I'd like to thank the organizers for having me, and thank you all very much for your attention. And should you have any questions, these are my contacts. And I really look forward to the discussion. Thank you very much.